Most experiments study plant microbe interactions by focusing on colonization, but our protocol is designed to look both at colonization and maintenance of the bacteria on the root. We can change the experiment to fit different bacteria and plants, and we can even test multiple bacteria in each 24-well plate.
To begin, use a standard hole puncher to create discs of plastic mesh. Collect the discs in a glass container and cover loosely. Sterilize using an autoclave set to a 20-minute dry cycle. Use flame-sterilized tweezers to distribute approximately 40 sterilized mesh discs in a single layer across the surface of previously prepared plant growth medium, agar plate. Place 2 previously sterilized seeds at the center of each mesh. Seal the plate with surgical tape and incubate for 2 to 6 days at 4 degrees Celsius in darkness to vernalize the seeds. Then, place the plate agar side down in a plant growth chamber for 8 to 10 days under short day settings of 9 hours of light at 21 degrees Celsius and 15 hours of dark at 18 degrees Celsius to germinate and grow seedlings. Add 1 milliliter of bacterial growth medium to each well of a sterile 24-well plate. To transfer the germinated seedlings embedded in mesh, use flame-sterilized forceps to gently peel the mesh containing 2